So today's talk, we're going to cover three primary topics: human PBMC engraftment profiles, and then we're going to get into modeling human cytokine release syndrome in a new in vivo assay that was developed by a group here at the Jackson Laboratory. And then we're going to bring those two together and talk more in depth about preclinical tumor efficacy with a wide range of therapeutics, whether that's monoclonal antibodies, bispecifics, and CAR T, and bring some data in relationship to cytokine release syndrome associated with that. So as usual, before I get started, a few things about the Jackson Laboratory and why we do these things. Our mission is to discover precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. And as a reminder, the Jackson Laboratory is a nonprofit research institute. So we're conducting grant-funded research, which is leading to many new discoveries. And obviously we offer education programs such as this, so that you can understand how to use these new discoveries in our research in your work. And so this leads to fantastic and wonderful new innovative solutions that we can provide to you, and much of which we'll be talking about and discussing in this presentation today. So the Jackson Laboratory originated in Bar Harbor, Maine, and we've been around for well over 90 years now, but we also have other facilities around the world. We have a facility in California that is our contract research organization, as well as a breeding and mouse distribution facility. And we also have a human genomics center in Farmington, Connecticut. We are currently working on and are distributing mice in China. And so we have a new wholly owned subsidiary that is being established in China. And very excitingly, just this week, we have announced that we are starting out a new venture in Japan to further expand our global reach. So we are truly a global influencing organization, and we're really excited about being able to help human health worldwide. Okay, so what I'm going to do is break down my talk today in 4 discrete chapters so that you can understand not only the models that I'm talking about, the research behind them, but also how you can implement them in your drug discovery program. And so in chapter 1, I'm really going to give you just a brief overview of all the different platforms that we have, and so you can be aware of what are some of the things that we offer. And then I'll really get into the science in chapter 2, really discussing the PBMC humanization of 2 different mouse models, how they differ, why they're important, and how they can be applied. Chapter 3 will be really the crux of the talk in terms of really talking about this new in vivo cytokine release assay that was developed, which is very exciting. And then bringing it all together in chapter 4 and combining all of these things together. So chapter 1, what are some of the different things that JAXT offers that you could make available to you? First, let me start and talk a little bit about humanization. It's a broad term, and it can refer to humanization in terms of the expression of human genes, and those human genes can be targets for discovery, or they can be the expression of growth factors that support the transplantation of cells. So in some cases, mice are just genetically modified for human-specific purposes. In other cases, they're expressing cytokines and things like that that help to support the engraftment of immune cells. And those immune cells can be derived from hematopoietic stem cells to lead to a more complete overall immune system biology, or in PBMCs, where you just have a subset of cells that you ask rapid questions, get rapid answers. So in the context of drug developers, and particularly those that are in monoclonal antibody discovery, these molecules are highly engineered, and they have an Fc tail. And the importance of that Fc portion of that molecule is it is recognized by neonatal Fc receptors, which lead to cycling through the endosomal compartment, which leads to maintaining the half-life. And each of these molecules are going to have different binding characteristics and different half-lives. And so we have a humanized platform where the mouse neonatal receptor is knocked out, the human is knocked in, and it models human half-life in a mouse, which can give you important information about dosing, candidate selection, and therefore understanding the dosing leads to information that can be helpful for safety and tox. Those same molecules can be tested out in either a CD34 engrafted platform or a PBMC engrafted platform, either with or without tumors, to answer a number of questions about efficacy and safety and mode of action and those types of things. And these are very versatile platforms, as you'll see as we go through the talk today, that can be modulated to answer your questions. So of course, it's not just monoclonal antibodies, it's also bispecifics and trispecifics. There's a huge number of configurations of these molecules. So if you've got a molecule that's like on the far left here, that's got like back-to-back binding sites, of course, it's not going to have a FC portion, so it really won't be applicable to the FcRn platform. But we do have these highly engineered bi and trispecifics that maintain that FC portion. And those can be monitored for half-life, but of course also be tested in these other platforms. And also, as you'll see today, we can use these platforms to look at CAR T. We've done a number of characterization studies looking at CAR T, and there's a number of published reports. And of course, models that support CAR-NK, CAR-M, and people are being very innovative in the new things that they are creating to fight cancer and autoimmune disease. And so it's a very exciting time in the engineering of these different molecules. And we can certainly help you out in terms of characterizing them and getting the information you need to move your platform forward. So chapter 2, let's talk about the immunobiology of the NSG versus a new model we call the NSG-MHC1/2 double knockout. So there's a number of things that influence the engraftment of human peripheral blood mononuclear cells. And this is data that was published in 2009. And it's a really great paper because it covers a lot of aspects of putting these cells in mice, the graft versus host disease that develops. Because remember, these are T cells that are going to be trained in the human thymus. So when they see mouse tissues, they're going to respond against them. And so there's a number of things that influence the engraftment and the rate and speed and severity of the development of the graft versus host disease. One is with or without irradiation. So without, you still can get engraftment, but you're going to slow down, and mice are going to survive a little bit longer. They're going to lose weight, but not as quickly. If you include irradiation, what you're doing is you're killing off some of the mouse immune cell population, which gives a competitive advantage for those donor and grafted cells. So they accelerate their expansion more rapidly. Those mice are going to develop GvHD more quickly with an associated weight loss. But there's also cell dosing that's very important. So a lower cell dose is going to lead to longer survival, and a higher cell dose is going to shorten that mean survival time. But what we're also finding out now that's not shown here is that there are very specific donor-to-donor differences in the rate of all of these responses. So if you're going to use a PBMC sample and each one is from a different donor, you need to characterize those samples because those are from genetically unique individuals trained on genetically unique set of antigen presenting molecules. So their ability to respond against the mouse is going to be different. The beautiful thing, though, is that it's very repetitive, kind of data that you can repeat. So once you have those PBMCs characterized, you can use that as a source of cells and repeat those studies and get the same kind of kinetics of engraftment. So what exactly are the cell types that are engrafted in these mice? You can use flow cytometry to collect blood samples post-engraftment, label them with antibodies that recognize cell surface molecules with fluorophores, and just figure out what these cell populations are. So you can tell the difference between mouse and human based on mouse CD45 versus human CD45. And what we're looking at here is 4 different donors, A, B, C, and D. These are all in irradiated NSG mice. And we're looking at 2 different time points, either 5 or 10 days post-engraftment. You can see that the human population expands within that time period very quickly. And then you can ask, well, within this total human population, what proportion of those cells are CD3 T cells, CD19 B cells, CD14 myeloids, and CD56 positive NK cells? And you can see that this is primarily a T cell expansion model. There are a few B cells that hang around even out to 10 days. There are some natural killer cells, but they will drop off pretty rapidly over time. And even though the kinetics of engraftment and the development of GvHD is different from donor to donor to donor, the cell populations that are present there are very much the same from one donor to the next. Okay. So if you go back to that paper I was mentioning a few minutes ago that was characterizing the PBMC engraftment, also in that paper, Dr. Lenny Schultz, who created the NSG mice, who's from the Jackson laboratory and working with Michael Bram at UMass Med, they are a team that have been working up these models, creating them, doing the initial characterization, and then they go over to our innovative group and we figure out ways to apply them to your research in more in-depth ways. But one of the things that they did is they generated class 1 deficient and class 2 deficient NSG mice and clearly demonstrated that this is a xenogeneic graft versus host disease model that leads to the expansion of these PBMCs. But they also had the foresight to say, well, if we knock out both class 1 and class 2 in the mouse, and we're still able to get good levels of engraftment, then we've got a mouse model in which we can do longer term studies. We can put the PBMCs in, we can put tumors on board, and we can do a wide range of things without the complication of graft versus host disease. So this is the model that we have now. This is the long nomenclature for it. There are multiple alleles that are knocked out, but we call it the NSG-MHC class 1, class 2 double knockout for short. So how do these two models compare in terms of putting PBMCs into these mice? What we're showing here is human cells from the same PBMC donor into both platforms. Green is the double knockout, blue is the NSG. And we're just looking at a snapshot 5 days after engraftment. There's human engraftment, and there's similar quantities of CD3 positive T cells, B cells, myeloid cells, NK cells, and CD33 positive myeloid cells as well. But what you can see here that is fundamental difference in terms of body weight and survival is that at about 10 days, the NSG mice are going to start developing the symptoms of graft versus host disease, one of which is losing weight, and there's a number of other clinical signs here. And those are indicators that the mice need to be sacrificed because they're developing severe graft versus host disease, and those animals need to be euthanized. If you look within the double knockout platform, there's one mouse out of the 5 in this particular experiment that started losing weight, had to be sacrificed. But overall, those mice maintain body weight and have longer term survival. Now, this is not a 100% lack of GvHD in this model. In other words, you can increase the cell dosage to a point, even with a double knockout, that'll recognize different tissues and molecules in the mouse that you'll start to see a shortened survival time. So all these things need to be titrated, but you can get good engraftment with long-term survival with these mice. So in summary of where we are so far, JAXT really has been doing PBMC engrafted platforms for a couple of years now, so we have extensive experience. I've talked to you about 2 different models, and as I go through, there are other platforms that can be PBMC humanized, but these are the 2 workhorse platforms that we use for most of our studies. Within the first 5 days post-engraftment, peripheral blood is dominated by T cells and has a low frequency of B cells and NK cells, so it's primarily a T cell modulation type of platform. The NSG recipients begin developing symptoms of GvHD after about 10 days through a xenogeneic recognition of mouse MHC molecules. So if you knock out class 1 and class 2 MHC molecules in the mouse, you can delay and reduce the severity of GvHD and have a platform in which you have longer term studies that can be performed in that setting. So chapter 3, let's focus on using the PBMC engrafted mice to model and screen in vivo immunomodulatory therapies for induction and for the suppression of cytokine release syndrome. So this is also a very versatile platform. So, but let's step back for a moment and go back to why we're thinking about cytokine release, why it's important, and also why did we need to develop a new platform? What's wrong with what's already out there and being used today? So a little bit of history. Back, many of you will probably be aware and remember that back in 2006, there was a company called TeGenero that had developed a molecule called TGN-1412. TGN-1412 is what is known as a structure that is a CD28 superagonist. It's a humanized monoclonal antibody, but the way it binds is it can bind and complex CD28 on the surface of T cells in a manner that can activate that T cell even in the absence of ligation of TCR and its interaction with MHC molecules. So it's a potent T cell activator, which is different than a conventional CD28 specific monoclonal antibody, which can bind that co-stimulatory molecule CD28, but typically needs to have interaction with an antigen presenting cell in the TCR or some other molecule that can bind the TCR and together work to activate those T cells. Now, unfortunately, what happened when this superagonist went into patients for the first time, all 6 patients who were healthy individuals went into severe cytokine storm and entered into organ failure, neurotoxicity, and a lot of very dangerous symptoms developed. Now, the other important aspect of this is this molecule was tested beforehand in what we call in vitro assays, where you put human PBMCs in a culture dish, you add the molecule, and you look for human cytokine release. And it was negative. It was also tested in non-human primates and found to be negative. And it also underwent some testing in some rat models and found to be negative. So it was tested before going in human. So this demonstrates the unreliability of the current platforms. Now, this is not just related to monoclonal antibodies. There are now things called bispecific T cell engagers that we'll talk more about momentarily that hopefully you're familiar with at this point. But these are molecules that pull T cells into contact with tumors, activate those T cells to try to eliminate those leukemias, et cetera. And now people are trying this out for solid tumors as well. But they also can develop a cytokine release syndrome that can lead to neurotoxicities. And this is the warning label that comes on a clinically approved Blincyto or blinatumomab. So these are real things that are known to be associated with these new immunological therapies. And so we needed a better platform that would more accurately and model these for clinical application. So a little bit more about what's actually happening and why perhaps some of these other platforms weren't working out as well, like particularly the in vitro assays. So this is a really great review, and I encourage you to take a look at it if you're not familiar with CRS. But there's a number of therapeutics that activate T cells through a wide range of different mechanisms that can lead to these activated T cells that, of course, release a number of factors like interferon gamma and TNF-alpha that lead to this cascade of pro-inflammatory response to recruit and activate other cells of the immune system. But it's not just the immune cells that are part of this cascade. There's also the endothelial cells within organs and tissues in the vascular structure that can also release cytokines, can release chemoattractants. And so it's the other cells in the tissues that are part of this cascade that are very important in this reaction and are required to better model the system. And that's why we started developing this in vivo system to include all these cells and compartments to better model what's happening here. Now, this is also not just happening in these immunotherapeutics, but it's also happening in infectious disease. Unfortunately, everyone's too familiar with SARS-CoV-2, but this is something to mention because what's happening here is the virus infects the lung, for example. You start this pro-inflammatory response. There are receptors in the vasculature as well. There's a lot of tissue damage. There's this large pro-inflammatory response. This could lead to vascular leakage. Your lungs fill up with fluids and you end up on a ventilator. So really, these things are very similar in their cascading response. So what's really wonderful is we do now have an assay that can be used to better model the system. We call it the in vivo induced cytokine release assay. We have a number of different platforms. It's PBMC based. It's a 6-day assay of T cell and natural killer cell related CRS responses. And there's a number of different configurations that we can set this up in, just a rapid screening, or as you'll see in a moment, we can look at tumor efficacy and CRS responses at the same time, which is very important because what we're finding is that not every therapeutic, just because it has a high efficacy, also has high cytokine release. We're starting to see molecules that have high efficacy and low cytokine release, and so that it can help you look at lead selection for those different molecules and the different characteristics. And we can include a number of different study readouts, not just human cytokine quantitation, but we can do the tumor efficacy as I measured, but also very importantly, look at the systemic response, body weight and temperature, disease index scoring, flow cytometry. Because in a cultured dish, and you see cytokine release, what does that mean? Is it an acute cytokine release? Is it a prolonged cytokine release? Is the acute cytokine release to the levels that lead to organ and tissue damage, or is it just a spike and it's going away and it's fine? The culture dish doesn't allow you to discern any of that information. So what does it look like? So here what we're looking at is two different donors, donor A and donor B. We're looking at the release of human TNF-alpha, interferon gamma, IL-6 or IL-10. And these are mice that were engrafted, and on day 6, they were treated with either anti-CD28, a TGN-1412 biosimilar, so it's the same exact structure recreated for these assays, and then looked at 1 or 4 hours post-treatment to look at cytokine release. And what we could see is there are donor-to-donor differences in the magnitude of response, and there's differences either 1 or 4 hours in the timing of the response overall. So these are known molecules that clinically have shown cytokine release, and they're showing a spike in cytokine release in this model as well. So we're replicating clinically observed cytokine release. There is humor donor-specific differences in drug-induced CRS response, which is also observed in the clinic. Not all patients always have a strong CRS response. These results are very highly repeatable for any given donor. In other words, once you've got a donor characterized, you treat with some of these known molecules, you repeat the assay, and you get very repetitive studies. So you can use molecules like this as positive controls. Here, we're just looking at a comparison with the NSG versus another mouse that I haven't talked about yet called the NSG-SGM3. The difference between these is that this mouse has been humanized by transgenic expression of human interleukin-3, GM-CSF or CSFF2, and KIT ligand. Now, this mouse was originally produced to enhance the engraftment of myeloid tumors, but it's also found that when you put a hematopoietic stem cell into these mice, it contributes to improved myeloid engraftment and expansion. But the other thing that's interesting about this platform is it's now known that GM-CSF can accelerate a CRS response. And people have published reports where they've suppressed GM-CSF and decreased a CRS response without impacting the tumor efficacy response, and that's been published. But you can see here by the expression of human GM-CSF, you can get a higher cytokine response. So this is the same donor engrafted into both of these hosts and treated with anti-CD28. Keytruda is a clinically approved anti-PD-1 monoclonal antibody, which is not particularly known for CRS responses. There's been a few documented cases, but it's really considered in our assays to be a CRS negative control. ATG is an anti-thymocyte globulin that's a polyclonal antibody. It leads to large-scale depletion of T cells and can lead to a very high cytokine response. And you can see that is shown here by the release of interferon gamma and IL-10 6 hours post-treatment. Using the SGM3, we've also done a TGN1412 dose response study. So in green here, a light green, darker green, and then very dark green, we've got low, medium, and high dose. We're looking at 1, 2, 4, and 6 hours post-treatment with these different doses. And we're looking at human interferon gamma, human TNF-alpha, and human IL-6. And you can clearly see that there is an associated dose response. As you increase TGN1412, you have an increase in cytokine release. So this is a drug-related response. So I've already mentioned the fact that there are donor-to-donor differences in response, and this is a study just to highlight that even further. Each one of these letters represents a different human PBMC donor, and there's 9 different ones here. These mice were treated with either OKT3, which is an anti-CD3. This anti-CD3 monoclonal antibody will lead to activation of the T cells and an activation-induced cell death and a pancytopenia. So it can induce a very strong CRS-like response, and as well as the regular anti-CD28. And what you can see is that there's low responders and there's very high responders. And if you look even more closely, the differences in response from donor to donor is going to be different based on drug treatment as well. So even with a very high OKT3 responder, there are low responders for anti-CD28. So the donor really does matter. And this is important because now we can use it to capture donor to donor differences in response, which is very important for clinical application. So what about a comparison of the double knockout, the class 1, class 2 double knockout versus NSG? We saw that we could get equivalent levels and cell types of engraftment, but can you induce a CRS type response in this platform? So what we're looking at is a comparison of those 2 different mouse platforms. They're irradiated, injected with the PBMCs, day 6, treated with Keytruda, Yervoy, which is ipilimumab, which is anti-CTLA4, and or OKT3. These 2 molecules are not known for high cytokine release clinically, and they didn't lead to a strong cytokine release response in these 2 platforms with this particular donor, whereas OKT3 did. But the other take home here is that the class 1, class 2 double knockout very much can lead to modeling of this CRS response in vivo as well. So I mentioned that now hopefully convinced you that you can model the CRS response. So in the context of things like infectious disease and things like that, what about the reversal of that CRS response? Can we also use the platform to ask questions about, well, if we could reliably induce the CRS response, can we use it as a platform for anti-inflammatories? And this is evidence to support that, yes, you probably can do that. So what we've got is 2 different donors, A and B, and we look at cytokine release either 2 or 6 hours later. There's actually 6 grafts in each one of these subsets. The first 3 are without Humira. Humira is an anti-inflammatory drug that is anti-TNF-alpha. So it's a monoclonal antibody that binds up soluble TNF-alpha. So the first 3 are without Humira and the second 3 are with Humira. And so these 3 arrows are indicating the suppression of TNF-alpha release post-treatment with either anti-CD28 or OKT3. So the drug very effectively binds up and eliminates human TNF-alpha that's released by these responses. But the other question is, well, what about other inflammatory molecules? So TNF is released very early. So if you suppress TNF-alpha, does it help suppress some of the other pro-inflammatory molecules? And the answer is yes. There is a decrease in interferon gamma in the Humira treated group in both of these different donors. But again, we're seeing donor-donor differences in the magnitude of response as well. So what about a comparison of this particular in vivo assay to the standard in vitro assay? So what was done, and more of this data is published in the manuscript that you can see at the bottom of this page if you want to see more data on this as well. So on the left here, we've got the in vivo assay with 4 different donors. The treatments are OKT3 and anti-CD28. Those same 4 donors were put in the mice in vivo under the assay conditions I've already explained. They were given to a separate laboratory to run blind an in vitro assay, treating with the same molecules, but also including isotype controls. And what was found is that there was a consistent cytokine release in vivo across those 4 donors, but there was one donor that did not lead to a cytokine release in vitro. So there was what we would call a false negative in response. And that was shown up in both interferon gamma and IL-10. There's also evidence of false positives in the in vitro assay. And these isotype controls shown here in gray led to a cytokine release response. In some cases, there was a very high magnitude of response. So again, this supports what has been reported in the literature that many of these in vitro assays can lead to false positives and false negatives. So overall, the in vitro cytokine release assays are very inconsistent. They really can lead to false negatives and false positives. And they also don't offer a dynamic range to discriminate low, medium, or high responders. The quantification of cytokine levels vary across platforms and are disconnected for potential clinical implications. As I said a moment ago, so you see a cytokine release. What does it mean in terms of its intensity, duration, magnitude? Is it leading to something that should be concerning in terms of organ and tissue damage and neurotoxicity? You don't really know the answer to that. So there's a lack of physiological conditions. The in vivo assay is really quite the opposite. It really gives you that physiological component and gives you multiple different readouts to really answer questions about what does that cytokine release mean to the organism, and is it really a toxic level of response? It also permits tumor engraftment to assess on-target toxicity and efficacy. If you go back to that Yi paper, you'll see that there's a number of other molecules that I haven't talked about that have been tested. And the Jackson laboratory has a large bank of curated PBMC samples that are pre-characterized and can serve as positive and negative controls for conducting these types of experiments. Okay, so in summary for chapter 3, the PBMC engrafted mice treated with agents known for inducing CRS show a strong release of human cytokines. The human PBMCs show donor-to-donor differences in severity of overall CRS response. Both the regular NSG and the class 1, class 2 double knockout show similar CRS response when engrafted with the same donor. The in vivo CRS assay generates more reliable and more reproducible results than the in vitro assay. We've gone back to the freezer and repeated some of these studies up to 5 times with the same agents, with the same donor, with the same platform, and got repeatable results. The in vivo CRS assay allows for safety evaluation of new immunomodulatory therapeutics, which can help you in your lead selection because each one of your molecules is going to have different binding characteristics, and the binding characteristics can influence the magnitude of the release of the CRS response. So we could also, since this is highly reproducible CRS responses with some of these known controls, we can evaluate novel anti-inflammatory treatments as well. So let's, in chapter 4, kind of bring this all together more. And one of the things I want to point out here with this diagram is this is a very, very versatile model. Now, most people have been applying a lot of these therapeutics to liquid tumors, but people are more and more trying to look at solid tumors. And the growth kinetics of those tumors are very different. And so therefore you need a modular platform that can handle those different growth characteristics. So in a platform where you're using B-cell lymphomas and other types of liquid tumors, you can irradiate the mice, put the PBMCs in first, and establish the PBMC engraftment, and then simply IV or IP inject your liquid tumors that are labeled with GFP or luciferase or something like that. You've got a co-grafted mouse, and then you can treat with your bispecific or monoclonal antibody. Now, what's different about solid tumors is their growth time is typically going to be longer, and you have to set up a number of mice, and you do either a rolling enrollment or you select a subpopulation that all has the same initial tumor volume. So what you do is set up the tumor engraftment of the solid tumor first, and then once those tumors get to the appropriate size, then without irradiation, you can inject your PBMCs and now get your dual engrafted model. So that's why I've got this circle, because it depends on what tumor you're working with and how you need to set that model up. Now, we can also do CAR T studies where you can engraft the liquid tumor right away and then just inject your CAR T to respond to it. And you can do similar types of things with solid tumors as well. Put the solid tumors on board first, and we're going into more in-depth studies with CAR T, including those kind of studies with more advanced humanized models as well. And we'll, in the coming weeks and months, we'll provide some more data for you on those platforms. So for those of you who are not familiar with bispecific T-cell engagers, these are molecules that have been highly engineered. They're taken from a monoclonal antibody that has the binding end, in this case in the examples in the next subsequent slides that I'm going to share with you, that are specific for CD3 that's found on T cells. And then typically another tumor-associated antigen, whether that's CD19, EpCAM, HER, EGFR, etc. And they're bound often back to back, or as I showed you earlier, they can have other configurations as well. But the data I'm going to show you in the next few slides has this back-to-back configuration. And what this does is pull the CD3 T cell into context with the target cell, and this binding leads to activation of the T cell, release of cytotoxic granules, and killing of the target. So what I'm going to talk about first is a CD19 CD3 BiTE, and we're going to compare this to rituximab. Rituximab is a clinically approved monoclonal antibody, which is an anti-CD20. So it's used for B-cell lymphomas because B cells express CD19, CD20, CD22, unique B-cell targets like that. And what we've done is we PBMC engrafted irradiated NSG mice, introduced the Raji B-cell lymphoma that was labeled with luciferase, so you can track these lymphomas systemically. And after treatment, ask questions about interferon gamma, TNF-alpha, and IL-10 release. So we've got rituximab alone, the positive control anti-CD28, the CD19, CD3 bispecifics alone, and then a combination treatment to see if a combination of rituximab and the bispecific together could enhance clearance of the tumor. And also, what would that mean in terms of cytokine release? This study was performed across 3 different donors, A, B, and C. Again, we see the donor-to-donor differences in response, with donor C showing the largest magnitude of response across all of these different cytokines that were released. And here we've quantitated the flux, meaning lower amount of signal means suppression of tumor growth. And you can see with anti-CD28 that didn't improve any response against the tumor by the PBMCs. And so there's continual expansion of this tumor in this mice. Rituximab suppressed tumor growth. So did the bispecific, but not quite as much as rituximab. And the combinatorial treatment was probably not statistically significantly improved compared to rituximab alone. But now if we go back and compare that to the cytokine release data, we can see that at least with some PBMC donors, we can see that the combinatorial therapy, even though it doesn't lead to improved efficacy, it does lead to higher cytokine release. So now you can design studies that can look at a comparison between efficacy towards the tumor and safety in terms of cytokine release. And as we do more of these studies, we're beginning, as I said earlier, to find studies where we can discern the difference between molecules that might have high efficacy and low cytokine release, which is obviously what you're going to be looking for. The other thing that's a key to these studies is the specificity of the type of response. In other words, in order to get T cell activation with a bispecific, you need to have engagement of that T cell with the tumor. So here's a study to demonstrate that. So in this case, we've got an epidermal growth factor receptor bispecific with CD3. We're looking at a triple negative tumor cancer cell line called MDA-MB231. These are cells that is a single cell suspension labeled with luciferase and IV injected into these PBMC engrafted mice. We're comparing Keytruda versus the bispecific, and then the EGFR CD3 bispecific in combination with Keytruda. Again, as a reminder, Keytruda is a checkpoint inhibitor. So it interrupts the PD-1, PD-L1 pathway, and it should enhance T cell responses. All right, so one of the things that's very important here is that in the absence of tumor, either 2 or 6 hours post-treatment, there's no activation of the bispecific. So when you do see cytokine release in the presence of tumor, you know that that bispecific is engaging the T cell is being activated, and you're getting an activated T cell response against the tumor. Now, the combination therapy did not, if you notice here, lead to an increase in cytokine response. But remember that piece of information as we go through the next slides. So what we're looking at here is the luciferase signal using xenogen imaging. So a decrease in signal is a decrease in tumor burden. An increase in a signal is an increase in tumor, either a broad expansion or a slow, steady expansion. So the slow, steady expansion is in the PBS and anti-CD28 control, Keytruda control, shown in green. Keytruda had no impact on tumor suppression at all. What's interesting about the anti-CD28 is it can lead to T cell activation, but it can also lead to T cell mediated activation-induced cell death and a cytopenia, a T cell cytopenia. So the lack of those human T cells led to tumor outgrowth. The bispecific and the bispecific plus Keytruda suppress tumor growth. This is the average data, and these are the individual mice. Now, what's really important about this in vivo model, as I pointed out earlier, is you can really ask more systemic questions about what's happening in the entire system. And you can use things like clinical score and mean body weight to find out more about what's happening in terms of the toxicity with these animals. Now, remember a moment ago, I said that the cytokine release was very similar between EGFR alone and EGFR CD3 plus Keytruda. But what's different here is you can see by looking at clinical scores that with the dual treatment, the clinical score were higher earlier and for a longer duration. And so there was some kind of increased toxicity to the entire system of the animal in vivo. Interestingly, using body clinical scores, you can see that this expansion of tumor led to a late development of higher clinical scores. Going along with that model that the higher tumor burden was leading to the animals becoming unwell. And also you saw this late drop off of body weight. But what's interesting also about the bispecific treatments is there was a decline in body weight, and they did seem to bounce back a little bit. And which is also interesting with the EGFR CD3 bispecific, those body scores decline and the mouse became more healthy and they started regaining body weight later on. So you can really see how the cytokine release influences the overall system. Now, in this last example here with the bispecifics, I really want to point out the importance of and how you can use the NSG-MHC class 1, class 2 double knockout. So far, we've been talking about primarily just the NSG mouse. One of the things that I said earlier that we want to do is to be able to do longer term studies. You want to do longer term studies in solid tumors typically. And so what we're doing here is set up a cohort of mice that were engrafted with a CDX, the MB231. They were engrafted orthotopically into the mammary gland. They were enrolled on study when they were about 180 to 200 millimeters cubed. Then these class 1, class 2 double knockout mice were PBMC humanized and then treated with an EGFR CD3 BiTE or cetuximab. Cetuximab being a monoclonal antibody that's specific for EGFR, which blocks angiogenesis, has some ADCC activities, those kinds of things. But look at the timeline and look at the measurement of tumor suppression. Tumor suppression was greatest in the bispecific. The cetuximab molecule had very little impact on the growth. But look at the duration of the study that went out to 40 days, and look at the maintenance of body weight. So these mice remain very healthy. So if we had performed the same study in a regular NSG, by 21 days or so, or even shorter, these mice would have had severe graft versus host disease, and the study would have been needed to be terminated. So this demonstrates that with the class 1, class 2 double knockout, we can do these long-term studies. Okay, in the last example here, we're going to talk about CAR-T mediated depletion of tumor cells. And just as a reminder, this is what we call autologous T-cell mediated therapy. And this is important to note because what we're doing is taking the patient's own T cells, genetically inserting this molecule with a unique receptor that's going to recognize a tumor antigen or some other cell population, activate the T cells to respond against those tumors. Now, because these have to go back into the patient, they need to be the patient's own T cells. Why is that important? Because these CAR T cells are not only going to be expressing this new chimeric antigen receptor, they're still going to be expressing the native TCRs from the human patient. So if you take them from a non-HLA matched different human patient and put them in, those cells could cause graft versus host disease. Similar when testing in mouse models, those human cells and those TCRs can respond against the mouse cells and tissues. So that's important as well. And then just a reminder of what these constructs look like. We've got the intracellular domain of the TCR, the CD3 ZAP70 complex, and the zeta subunits. These zeta subunits can also be included with CD28 or 41BB co-stimulators, but they're attached or linked to these specific binding sites for your target of interest. So they're kind of a fusion molecule that's genetically expressed in these T cells. So here's just one example study where we took a human T cell population, transduced to express a CD22 specific CAR T. CD22 is also found on Raji cells, which are a B cell lymphoma. This B cell lymphoma is labeled with luciferase and GFP. So if you just inject these into the NSG-MHC class 1, class 2 double knockout, as you see over time, an increase in signal, that's an increase in tumor burden or the expansion of that lymphoma in the mice. If you treat with a mock CAR T, you want to do a mock CAR T because you want to control for those native T cell receptors. Are they responding against the tumor? Because this is a human T cell responding against a human cancer cell line that is not HLA matched. So you want to discern the difference between your target CAR T and just those native TCR receptors. And what we see is only with the CD22 CAR T do you see a depletion of those tumor cells over time. And this is just a look at the same data shown graphically. And we can see that the PBS and the mock CAR T had no impact on tumor growth. It's only the CD22 CAR T that suppressed the tumor growth in both the dorsal and the ventral positions of looking at these mice. And if we look at body weight, we can see that the CAR T maintained body weight, which means two things. we're getting rid of tumor burden, which could lead to decrease in body weight. And because we're using the NSG-MHC class 1, class 2 double knockout, none of those T cells are responding against host tissues, so those mice are remaining healthy. It's only in the context of tumor burden that we're seeing weight loss with the PBS in the mock CAR-T. So the summary of chapter 4, human PBMC and grafted mice co-transplanted with human tumors allow preclinical evaluation of human target-specific bispecific antibodies. The bispecific antibodies show donor-to-donor differences in cytokine release, and cytokine release occurs in the presence of tumor. You need to have tumor present to get that response. Tumor cytotoxic response is associated with T cell activation, mild cytopenia, and high cytokine release. Those are two aspects that I didn't have time to show you today, because what you can do that I haven't showed you is use flow cytometry to look at the things like mode of action. Are your T cells upregulating CD69 and things like that? Have you induced those T cells to the point where there's activation-induced cell death and those kinds of things? Bispecific induced CRS increases body scores indicative of a systemic toxicity. The class 1, class 2 double knockout mice enable the preclinical assessment of novel human CAR-T mediated therapies in the absence of acute GvHD and allow you to do much longer term studies to collect more data. So in conclusion, JAXT has developed a novel humanized mouse model for testing CRS. It's rapid, it's sensitive, and it's very highly reproducible. It's rapid in the sense that it can be done in a quick 6-day assay, and it's modulation of T cells and natural killer cells. So you can use it as a screening tool for a wide range of candidates. We have presented to you single agent CRS data for OKT, anti-CD28, TGN1412, ATG, checkpoint inhibitors, bispecifics, etc. So it's a wide range of molecules that are really mimicking what we're seeing in the clinic as well. Importantly, there is donor-to-donor variability, which is evident in response to single agent and drug combinations. The bispecific antibodies can induce cytokines in the presence of tumors similar to or higher than CD28 and result in toxicity in the mice. And we have more data that we're collecting on organ and tissue damage as well. And so get in touch with us if you'd like to see some of the examples of that data. We have developed a translational humanized mouse model for preclinical assessment of CRS adverse events to monoclonal antibodies and bispecific antibody therapeutics. And now I'd like, as one of my last slides here, is I'd like to bring out a poll question. And please help me answer this question. And this helps me understand that I was able to convey the information that I wanted to you today. And the question is, when performing analysis of new immunomodulatory therapeutics, whether that's monoclonal antibodies, bio trispecifics, CAR-T, et cetera, why would you want to apply measures of in vivo cytokine release into the evaluation of your drug's performance? Is it to help with lead selection in early development process? Is it to help you understand the safety profile of your drug and assist in selecting dose? To help you anticipate the range and severity of toxic CRS across the genetically diverse population? To obtain more accurate and reliable data than what can be obtained from in vitro assays? All of the above, none of the above? Let me know what you think the answer is to this. All right, getting late in time, so I guess we should post the results. And of course, the answer is all of the above. It's definitely the best answer. So it's a unique platform that can do quite a range of different things. So I do need to acknowledge the people who did this work. I just have the pleasure of presenting this information to you. Of course, we've got the JAXT product development group that developed this data, but that data could not be developed without the help of Lenny Schultz creating these models and our collaborators at UMass Med. who really worked out the initial characterization and passed it over to our team at JAXT. And just as a reminder, we're not just a research institute, we're not just a repository and distributor of mice. We have a wide range of service offerings. So if you have questions about things related to mice, please get in touch with us. And thank you very much for your attention today.